Good afternoon. Welcome back, folks. It's my pleasure this afternoon to introduce to you Patrick Geisler, Associate Professor of Anthropology in our Sociology and Anthropology Department at Gustavus here. And he will introduce our next speaker, Professor Ramachandran. Patrick?
Good afternoon, everybody. Welcome back. What we turn to now, I got the first one, good. What we turn to now are some of the most mysterious, puzzling, or exceptional human experiences, which nevertheless can shed tremendous and unexpected light on the normal brain and being human. Many such experiences or conditions are described in the medical literature, but are not explained or resolved. Take, for instance, the puzzling experience of an amputee named James, who complained of excruciating pain from clenching his missing or phantom right hand so tightly that his missing fingernails dug into his missing right palm, as James tries to show us here. How in the world could that be? If you're a doctor, who would you call to unclench a fist that isn't there? Relieve the terrible pain and explain this centuries-old medical mystery of phantom limbs and phantom pain, something experienced by a majority of amputees. Or what about David? Survived a car accident and seemed perfectly normal mentally and in his interactions, but insisted oddly that his parents, as well as his dog, were just imposters. Or then there's John, the young man with temporal lobe epilepsy, who professes deep religious-like experiences of God, oneness with the world, and other mystical perceptions during his seizures. What's going on? What might such seizures, which are the overstimulation of certain networks of neurons in the temporal lobe, have to do with these intense religious experiences? Again, for these and so many other strange medical anomalies, who do you call? You call our next speaker. Dr. Vilayanur S. Ramachandran, who is often referred to as the Sherlock Holmes of neuroscience for his inspired medical sleuthing and what it reveals to him about previously unknown mechanisms of the normal brain and its relationship to the mind. In this case of James's clenched phantom fist, for instance, Ramachandran deduced that the brain was not getting the necessary sensory feedback from a real hand that would say, hey, stop clenching, and then one would stop immediately. To prove it, Ramachandran supplied that needed sensory feedback, but visual sensory feedback. He used a mirror box he created to give James the illusion of having two hands. When James saw two fists in the box, his good left hand and its mirror image exactly where he had placed his phantom right hand in the box, he immediately opened them both, and years of phantom pain disappeared. Although Ramachandran is well known for his medical sleuthing and ingenious, low-tech solutions to neurological puzzles, he is also well known for his research and theorizing about several other frontier areas, such as the construction of a body image in the brain, the role of neurons, mirror neurons, in cultural transmission, evolution, consciousness, and the brain's plasticity, or that is, that previously unheard of and unimaginable ability of the brain to rewire itself after an injury, something revealed to him and confirmed in his investigations of amputees' experiences. Ramachandran was born and raised in the state of Tamil Nadu, southeastern India, and even at an early age was attracted to research and discovery. Both at his father's urging, he completed an MD at the Stanley Medical College in Madras, or as it's called today, Chennai, though he was soon drawn back to research and pursued and obtained a Ph.D. in psychology from Trinity College, University of Cambridge. Today, he is Distinguished Professor of Psychology and the Neurosciences program and Director of the Center for Brain and Cognition at the University of California, San Diego. He has published several best-selling books and over 100 papers in scientific journals and boasts a long list of honors, including the Adriaanse Kappers Medal from the Royal Netherlands Academy of Science, a gold medal from the Australian National University, and the Presidential Lecture Award from the American Academy of Neurology, just to mention a very, very few. PBS devoted a NOVA program to his discoveries, as did Time and the BBC. And both Newsweek and Time magazine named him one of the 100 most important or most influential people in the world. And I know that he is that influential. I know it, at least in part because he's such an extraordinary communicator in his books, in these programs, about the complexities of the mind and being human. I assign his classic, Phantoms in the Brain, to my first semester students in a first-term seminar I call Exceptional Human Experience. It floors them, and they talk about Ramachandran and his work for years. One of those students was Ross Puffer, who graduated from Gustavus a few years ago. He's now in his third year of medical school, doing rotations at the Mayo Clinic in Rochester, and is aiming for neurology and neurosurgery at the Mayo because of that early inspiration by Ramachandran. In fact, he is here today because he heard Ramachandran would be speaking. So Ross, I won't let you wait any longer. Please welcome Dr. Vilayanur Ramachandran.
Well, thank you very much, Dr. Geisner, for that amazing introduction. First of all, they say you should never begin a talk with an apology, but in this case, I'm going to make an exception. I'm deeply honored by the Gustavus Adolphus College, the Nobel Foundation, and Dr. Niederreiter for inviting me to give this lecture today, but can't be there in person because of unexpected circumstances here in La Jolla. And it sort of makes me feel quite awkward. And I think it's slightly disrespectful to the audience, but we'll see how it goes. As you just heard, I do research in behavioral neurology, and we can approach the structure and functions of the brain from many different angles. You already heard an introductory talk this morning, so I won't go through the basics, but brain consists of something like 100 billion nerve cells, and each nerve cell makes contact with adjacent nerve cells at synapses. There is about 1,000 to 10,000 points of contact between adjacent neurons, and each synapse where exchange of information occurs can be either on or off, can be inhibitory, can be excitatory, can be on, let's see what else. Inhibitory, excitatory can be on or off. And based on this information, someone has figured out that the number of possible brain states exceeds the number of elementary particles in the known universe. This gives you some idea of the complexity we're dealing with here in studying the brain. Someone once asked me the other day, a reporter, is it true that we only use 1% of our brains, Dr. Ramachandran? And I said, there's no truth to this at all, except in Texas. So, how do you study the functions of this organ? There are many different approaches. You can do functional brain imaging. You can record from individual nerve cells, eavesdrop on the activity of nerve cells in the brain. Or the third method, the one we use, is called behavioral neurology, or sometimes called cognitive neuroscience, or one aspect of cognitive neuroscience, where you look at patients who have sustained an injury or a genetic change in a small region of the brain. When there's a change in a relatively tiny part of the brain, what you get is often not an across-the-board blunting of your mind, complete reduction of your cognitive abilities. You get a highly selective loss of one function, other functions being preserved intact. This gives you some confidence in asserting that that region of the brain is somehow involved in mediating that function. See, I'm going to show you several examples of this in today's lecture. It just shows you a neuron. So what we do is we take patients who display very odd, unusual symptoms. Many of these syndromes, like Apotemnophilia, which I'm going to tell you about, have been known for ages, or maybe over 100 years, but have been brushed aside as curiosities or anomalies, which people couldn't make any sense out of. A lot of the time, you can't make any sense out of them. but sometimes you hit the jackpot. You develop a nose for anomalies, and every now and then you get lucky and you find some syndrome or disorder that gives you key insights into the functions of the normal brain, not only explaining the patient's syndrome, but explaining how your brain and my brain works. So one classic example of this we stumbled on not long ago, maybe a year ago. I'm going to talk a great deal in this lecture about phantom limbs and also about synesthesia, but this is work that's more than a year or two, started more than 2 or 3 years ago. It's already familiar to you, so I'm going to start with something which is quite new. This is a disorder called Apotemnophilia. It refers to people who are otherwise completely normal. For example, we saw the dean of an engineering school recently, who soon after retirement claimed that he had a lifelong urge to have his own left arm amputated just below the elbow. So this is the converse of phantom limb. Phantom limb patient has lost his arm and continues to vividly feel the presence of the arm. Here you have a person who has a normal arm, no pathology apparently, who wants to have his arm removed. And many of these people claim that they have this urge to have one arm removed or one leg removed from as far back in childhood as they can remember. And this desire for amputation is called apotemnophilia. And for a long time, people ascribed it to psychological reasons, including Freudian, all sorts of Freudian explanations have been proposed, including the somewhat absurd view that the patient wants to draw attention to himself. So he goes through the procedure of having it amputated. By the way, 1/3 of these patients subsequently get themselves actually amputated. And this relieves their depression, and they feel much better as a result. So this is not something trivial and unimportant clinically. It's not rare. You see there's a website devoted to apotemnophilia. And then of every 10 apotemnophiliacs, 3 or 4 actually go get themselves amputated. It's not legal in this country. They go get it done in Mexico, I believe, or Canada sometimes. So why does this happen? Well, one theory is they're trying to draw attention to themselves. This doesn't make any sense. Why would you go through the drastic step of having an arm amputated just to draw attention to yourself? It didn't make much sense. Why not get your nose removed or one finger removed? Why an arm or a leg? Seems quite drastic. And the second explanation, oddly enough, is a Freudian one. These people have their arm amputated in order to develop a stump which resembles a giant penis. Now, I'm not making any of this up. You can go into the internet and you'll find this explanation. It's actually been proposed as an explanation for apotemnophilia. So all this is clearly nonsense, but the question is what actually is going on. So if you look at the brain, the side view of the brain, I understand this audience has a lot of lay people, so I'm going to say some very basic things and hope I don't insult the experts. That's the central sulcus, or also called the Rolandic sulcus, big fissure running through the middle of the brain. Behind it, see this cursor is working. Behind it, that vertical brown strip is the postcentral gyrus. All of this is in the parietal lobe of the brain, on the side of the brain. In the postcentral gyrus, there is a complete map of the surface of the body on the surface of the brain. So every point on the body surface is represented on a point on the surface of the brain. There's a complete map of the body draped on the surface of the brain. There's certain peculiarities in this map which we won't go into now, but I will talk about when we get to phantom limbs. But for now, all you need to know is there's a complete map. In fact, there are several maps, S1, S2, and so on and so forth, but I'm going to talk about just the basic S1 map in the brain, with every point on the body surface being represented at a corresponding point on the surface of the brain. Now, from this region of the brain, messages go to S2, behind it, and from there it goes to an area called SPL, superior parietal lobule, where you construct your body image. If I shut my eyes, I have a very vivid sense of different parts of my body, and I open my eyes, of course, my vision confirms the existence of the body, different body parts. Then if I shake my head, there's vestibular sensations coming in. So, and then, of course, there's auditory space, and all of this converges on to the superior parietal lobe to construct what you call your body image. Henry Head and Lord Russell Brain coined this phrase, body image. There's a very vivid sense of your own body your own mind occupying your own body or whatever. So now, it turns out, so we said, well, maybe what this guy is suffering from is maybe his arm is intact and normal and sending signals, but when the signals go to S1, somatosensory cortex 1, the map in the postcentral gyrus here, maybe in that map, the hand is not represented anymore, or the arm is not represented. Therefore, there's an alienation of the arm. The patient says, the arm feels like it doesn't belong to me, it's abhorrent, I want it removed, and so on and so forth. But when we ask these patients carefully, I don't like to call them patients, by the way, because they don't necessarily regard this as abnormal, but I'll use it as shorthand. So if you ask them, they'll say, I asked one of them, for example, does this arm feel like it's intrusive, or does it feel like it doesn't belong to you? The patient clearly says, no, it belongs to me all right. It just feels like it over belongs to me. It's intrusive. It intrudes on my consciousness, and I want it removed. And then my postdoctoral colleague, Paul McGeough and David Brang, a grad student in our lab, decided to explore this phenomenon further and see if there might be a neurological basis. Another reason for believing it's not psychological is, if you give the guy a felt pen, he'll take the pen and draw the outline, often an irregular outline, at a specific point on the arm, below which he wants the arm removed. And if you come back after a few weeks and ask him to redraw the line, he'll draw it exactly along the same place, even though you could not have possibly memorized it. This suggests strongly that there might be a physiological basis for this. And our first hunch was that this could be, the arm representation could be missing in the somatosensory cortex, maybe in S1, maybe in S2. So how do you test this? Well, we went and did a galvanic skin response. If you touch somebody on different parts of the body, you say poke him with a needle, the message goes to the somatosensory cortex, then goes to the amygdala, goes to the insular cortex, goes to the amygdala, where you pick up these pain signals and then convey them to the anterior cingulate, or eventually down into the hypothalamic nuclei, cascades down the autonomic nervous system, making you sweat, preparing your body to fight and flee and have sex, making you sweat, right? If you can encounter anything that's salient or irritating or annoying or something you're to be scared of, you believe it or not, your skin starts sweating immediately. You can measure the sweating using 2 electrodes placed on your palm to measure the change in skin resistance. This is called the galvanic skin response, or skin conductance response, GSR. So when you and I look at a piece of paper, or if somebody caresses you, you're not going to get a GSR. But if somebody pokes you with a needle, you look at a lion or a tiger, you get a big GSR, galvanic skin response. So this gives you a gut level, visceral, sensory reaction to something you're looking at, whether it's arousing or fearful or something of that nature. You start sweating and the sweat is measured. And this is involuntary, you can't control your galvanic skin response. That's what makes it an advantage. Instead of just asking the guy, do you feel bad, you can actually measure his SCR and monitor his emotional reactions. So what we did was, we drew the line with a felt pen, then erased it, of course, and we touched different parts of the body, poked different parts of the body with a needle, and then measured the galvanic skin response on the other hand. If you poke the skin of the hand or the arm below the felt line where he wants the amputation done, you get a huge big jolt in his galvanic skin response. If you do it above that, there is no jolt. If you do it in any other part of the body, there's no jolt. So clearly, stimulating him below the line of amputation, where he wants his amputation done, then the discrepancy is picked up by the brain and experiences an aversion, which you see as a galvanic skin response. This shows you direct physiological evidence that this is a genuine physiological, neurological phenomenon, not some psychological phenomenon. Right? So how do you prove this further? Well, we did the obvious thing of brain imaging, which we sometimes use. And we wanted to see if there's a misrepresentation of the arm in S1, and found, in fact, S1 map was completely normal. The map of the body surface on S1 is completely normal in this chap. But the message, when you go to SPL, superior parietal lobe, and touch different parts of the body, and measure activity in SPL, you get normal activity from different parts of the body. But when you go below the line of amputation and you touch him, there is no response from SPL. This shows that even though the sensory signals are coming into S1, so sensory regions of the brain, somatosensory cortex, and possibly into S2, the messages don't have a place to get to because the arm is missing in the representation of the body image in SPL. So this creates a acute discrepancy in the brain. Exactly how and why is not clear. But the discrepancy probably then goes and gets picked up by the insular cortex, and then the messages get sent to the anterior cingulate, cascading down the hypothalamic nuclei, down the autonomic nervous system, and already producing the GSR, as I said, and also producing a lack of response in the brain imaging when you look at SPL. So here's an example of a bizarre, seemingly incomprehensible neuropsychiatric disorder, or psychiatric disorder, Apotemnophilia, where you can discard the Freudian explanation, come up with a physiological account of what's going on in the brain, test it using galvanic skin response brain imaging, and show that you're probably on the right track. And incidentally, it's told you something about how the normal brain constructs body image. Now, I want to add a note of caution. Unlike the remaining two segments of my talk, which is going to be about synesthesia, about phantom limb, this is very early work in progress. It's just been published. So I'd add a note of caution saying it needs to be replicated by other groups before you can accept it as being a valid explanation. But we think we're on the right track. So next I turn to phantom limbs. Some of this has been widely publicized, over-publicized maybe, but I'll just run through it briefly. You all know what phantom limbs are. Arm or a leg is amputated. You continue to vividly feel the presence of that missing arm or leg. And many people experience, if you, just out of curiosity, inspired by some animal experiments, I took a Q-tip and touched different parts of the body of a patient who had lost his left arm in an accident, experiencing a phantom left arm. And up to 3 weeks after his amputation, he came to my lab, and I had him blindfolded and touched different parts of his body with a Q-tip and asked him what he felt. So his left arm was amputated below the elbow, and I touched different parts of his face, left side of the face and right side of the face, and chest and neck and forehead, with a Q-tip, and asked him, "Derek, what do you feel?" And he said, "Well, you're touching my forehead." "Do you feel anything in your phantom?" "No, absolutely not." "How about here?" "Oh my God, I feel that on my cheek, but I also feel it on my phantom thumb." "How about here?" "That's my phantom index finger. That's my phantom pinky." In fact, there was a complete map of the missing phantom hand on the surface of the face. That's the thumb, index finger, pinky, ball of the thumb. And the boundaries between the different individual digits are clearly demarcated, so it's almost like you're plotting receptor fields. And it turns out there is another map. So first of all, why does that happen? Why would it be the case that you take a patient with a phantom limb, phantom left arm, and you touch the left side of his face, he experiences sensations as emerging from his phantom hand? These are topographically organized. Well, it turns out, as I said, there's a complete map of the body surface and the surface of the brain. Cursors. Okay. Microscopic cursor here. I hope all of you can see it. There's a Penfield map. You can see it's a complete map except for certain peculiarities. That is, the hand representation, the representation of the head are right next to each other. In other words, the head, instead of being near the neck, is dislocated and below the representation of the hand. So our reasoning is, when you amputate the arm, sensory signals don't go from the arm. There's no sensory signals coming to the hand representation of the brain. So it's hungry for new sensory input. The sensory input from the skin of the face normally only goes to the face area, now invades, so to speak, the missing, the hand area in the brain that's been deafferented and doesn't have any signals coming in. I hope that's clear. So the hand area does not get any signals. Sensory input from the face, instead of just going to the face area as it normally would, now innervates the cursor doesn't work. Sorry. Invades the territory that originally corresponded to the missing hand. And these signals, or this activation of the hand area, is then misinterpreted by higher centers in the brain as arising from the missing phantom hand. This distance is over a couple of centimeters, so this is quite radical. We're showing in a matter of weeks, and now we've shown this happens in as little as a week or a few days. You get a complete reorganization of the Penfield map in S1. So we all grow up as learning medical textbooks or psychology textbooks about the Penfield map in the brain. What these experiments are suggesting, these maps are actually dynamic mosaics of activity representing the sensory surface. And often when you remove an arm or a leg, there's a shift in equilibrium rather than a complete loss of signals and a degeneration of that region corresponding to the brain. Now, it turns out there's another map here on the, just above the stump, which I won't go into. You can do magnetoencephalography. This is Tony Yang, who's a medical student working in our lab, maybe about 15 years ago, showing the reorganization in the cortex. Now, the other thing we noticed is many patients with phantom limbs have painful cramps in the missing phantom arm. And very often these are patients who had a brachial avulsion, so the intact arm was actually paralyzed and lying in a lying in a sling for several weeks or months prior to the amputation. So there was a painful intact arm lying in a sling for several months, and to get rid of the pain, maybe in a misguided attempt to get rid of the pain, the arm was amputated, and the chap was then left with a phantom arm lying in a phantom sling with phantom pain. And often the phantom is frozen in the same position it was in when it was paralyzed. So when you amputate it, there's a paralysis of the real arm is in some sense carried over into the phantom. Now, we have some thoughts about why this might happen, but I'm going to skip over all that given our time limits. But many of these people have excruciating phantom pain in the arm. It's often in a very awkward, clenched position. They'll mimic the position of the phantom with the real hand, saying, "Doctor, it's like this," pointing upwards to the ceiling, and it's twisted like this in an anatomically impossible position. In fact, the wrist is facing upwards and dorsiflexing, almost having the fingers touching the back of my wrist. It's anatomically impossible, but that's what the phantom is doing. It's excruciatingly painful. I wish I could move my phantom. If I could do that, it might relieve the pain. So that's when we hit on the idea of using the mirror box. So you have a vertical mirror propped up in front of the patient in the cardboard box. Patient puts the right hand on the right side of the mirror, looks at the reflection of the right hand in the mirror, and sees the mirror reflection of the right hand superimposed optically on the felt position of the phantom limb. So now you've resurrected optically his phantom. If you now send signals to both hands simultaneously, as shown in the slide, both hands simultaneously, like clapping or conducting an orchestra, so long as they're symmetrical movements, he's going to get the visual illusion that the phantom hand has been resurrected, but also is obeying his motor commands. So now he's getting visual feedback that the command to the phantom is being obeyed. What happens then? This mere act of restoring the feedback loop through using vision seems to animate the phantom. So the first time in the 10 years, the patient's arm has been paralyzed and fixed in a position for 10 years and is excruciatingly painful. For the first time in 10 years, the guy can actually start moving his phantom. Not only does it look like it's moving, what's critical is it actually feels like it's moving, and this animates the phantom and liberates the phantom from its pain. And this often happens instantly as soon as you do the experiment. And if you continue the procedure for several weeks, often the patient's pain, phantom pain, goes away completely. And each patient is his own control because they've been through morphine, been through all kinds of medications, sometimes dorsal trisotomies, often removing neuromas to no avail. Then you put a simple mirror and the mirror works, so it's not really likely that it's a placebo. But having said that, it's important to do these experiments. There was a randomized trial done by Tsao's group in Walter Reed, and they did experiments where they had phantom limb patients, about 22 patients, I believe. And is the cursor again? Control group of 16 patients. 8 patients had a covered mirror. 8 patients uses mental imagery, no mirrors, to try and move or animate the phantom limb to relieve the pain several times a day. And the experimental group had a mirror, and from a pain of about, on a scale of 1 to 7, from a pain of 4 or 5, the mirror group dropped down to, from 40 or 50, dropped down to about 5, all the patients. The covered group showed, if anything, an increase in the pain to about 6 or 7 on a scale of 5 or 6 on a scale of 7. And the mental visualization dropped only slightly, but not significantly. And then when they were crossed over after 4 weeks, all of them dropped down to a level of about 1 on a scale of 7, where the pain was barely noticeable and certainly quite tolerable. So this is very striking evidence, and there have been a number of studies showing this, and these mirrors are now used throughout the country, indeed throughout the world, as a partial treatment for phantom limb pain. I'd like to tell you about another extraordinary condition called RSD, where the issue is not phantom pain, but a real arm with a trivial injury. And believe it or not, when there's a trivial injury, ordinarily the injury heals and you recover from the pain after a few weeks. That's a normal course of events. But sometimes the pain persists with a vengeance. You have a trivial injury like a metacarpal bone fracture, and the hand is temporarily paralyzed as a result of excruciating pain. But instead of the hand healing after a few weeks, instead of pain going away, the pain persists with a vengeance. And in fact, the bone starts atrophying, the hand starts atrophying. You get warmth and you get excruciating chronic pain. The pain fails to subside. And the hand becomes completely paralyzed and swollen and becomes inflamed, all starting from this trivial injury to the metacarpal bone. Long after the healing of the bone, you get inflammation, swelling of the arm, and spreads to include the entire arm. There's pain, there's immobilization, there's swelling, an atrophy of the bone called Surex atrophy. This is called RSD, reflex sympathetic dystrophy, or complex regional pain syndrome. Nobody knows why it happens, but I thought maybe it's a form of learned pain. So every time the patient tries to move his hand during the acute phase of the pain, the signal's coming in saying, ouch, don't move. So every act of trying to move the hand, he gets visual signals saying that the hand's not moving, and it's excruciatingly painful. This somehow goes awry, and there's a Hebbian link between mere attempt to move the hand, and the pain signals coming in. So the brain just gives up, and you refuse to move the hand, and it completely gets paralyzed. So we call it learned paralysis. The patient then gets stuck with this learned paralysis. Any attempt to move the hand, long after it's healed, generates excruciating pain. It's not rare. 1 out of 10 stroke patients develops this after stroke. And after a trivial injury to the finger or skin, maybe 1 out of 30 or 40 patients experiences this. And the condition is lifelong and there's no known treatment. So we said, let's try a mirror just for fun. Maybe you can unlearn the learned pain, showing him that he can move his paralyzed, painful arm with impunity and there's no pain, because it's really mirror reflections, it's moving. So we put a mirror in the box. Let me take that back. We didn't do this experiment. It was done by Candy McCabe in England, in Bath, actually. She and her colleagues, and along with Peter Halligan. And Patrick Wall was one of the co-authors, who was a world-leading expert on pain. He was very familiar with the idea of placebo, so I was confident that his result is certainly likely to be a valid result. Put a mirror there, and the normal hand is put on the right side of the mirror. The painful, paralyzed arm is real arm. We're not talking about phantoms now, but by real paralyzed arm, put on the left side. He moves the normal right hand, looks in the mirror, and it looks like the paralyzed, painful arm is moving. Astonishingly, the pain subsides even as he's watching. The inflammation starts subsiding. You can measure with the thermometer, the temperature changes as he's watching with the mirror. So these are actual physiological changes produced in the living, paralyzed arm, which you absolutely cannot fake. And as I said, one of the authors is Pat Wall. So this convinced me that you're dealing with not only phantom pain being relieved by visual imagery from a mirror, but real pain in a real arm being relieved. And I used to think that mind-body medicine is all mumbo-jumbo, but seeing this convinced me of the reality of visual signals influencing pain in a real arm, changing inflammation, changing swelling in a real arm. One last point I want to make about phantom limbs, and then we'll let that go. And that has something to do with mirror neurons. And I have to say something about mirror neurons, obviously. I think most of you here know what a mirror neuron is. They were discovered by Giacomo Rizzolatti in a region corresponding roughly to the Broca's area in the human brain, in the frontal lobe. And what they found was in the premotor cortex, there are cells. We know about motor command cells. We've known this since the time of Vernon Mountcastle, from 50 years ago. There are cells, let's take my right frontal region. There are cells in the Broca's area, the vicinity of the Broca's area, which send signals out, they're called motor commands, which go and command my hand to reach out and grab a peanut. So a set of signals that orchestrates a precise sequence of muscle twitches to grab a peanut originates in my frontal lobe. And then I reach out and grab the peanut. Or there's another neuron which fires whenever I reach out and pull a lever, another one for pushing a stone, another one for putting something in my mouth. Right? These are called motor command neurons, they're well known. These are your garden variety motor command neurons. But a subset of these neurons, called mirror neurons, it turns out, will fire when I merely watch one of you in the audience reaching out and grabbing a peanut. So if I then go to the lever pulling neuron and record from that, it fires when I go and pull a lever, but it will also fire when I watch you pulling a lever. These are promptly dubbed monkey see, monkey do neurons because they were discovered in monkeys, or mirror neurons since then because they've also been found in humans. So when I heard about this first time, and I'm sure hundreds of people had the same reaction, we jumped off our seats saying, my God, here's a neuron that's reading another monkey's intentions. Because what this neuron is saying is, in effect, hey, look, the same neuron is firing as would fire if I were to reach out and grab a peanut. Therefore, that person is about to reach out and grab a peanut. So it's performing a simulation, virtual reality simulation, of the other person's mind and mental propensities, and maybe involved in constructing a theory of other minds. Now, this is well known, but there's another group of mirror neurons called sensory mirror neurons, discovered in the region I alluded to earlier. Roughly in S2, these neurons respond ordinarily to touch signals. If I touch you, neurons in your S1 and S2 fire. So these are sensory touch neurons. But a subset of them, maybe about 10% of them, will fire if I watch you being touched in the corresponding point in the body. So this neuron fires when somebody touches me in my somatosensory cortex, but it will also fire when somebody touches you in the audience and I'm watching you being touched, right? So again, here is a mirror touch sensory neuron instead of mirror motor neuron, which is in a sense allowing me to create a virtual reality of your sensation, saying the same thing is happening to me, same thing is happening to him as would happen if I were touched. So he's being touched, I can empathize with your touch sensations. Now this raises an interesting question. If this is really true, the same neurons are firing or a subset are firing, then when I watch you being touched, the sensory neurons are firing, I don't say ouch. When somebody pokes you with a needle, I empathize, but I don't say ouch. I don't literally experience the pain qualia. How come? How does the brain know the difference? Well, it turns out one reason might be is inhibition from frontal structures, but another reason might be there's signals from your intact hand, from your skin receptors, going back to the brain, informing the brain, including S2, saying, look, you're not being touched. So there's a veto signal saying, look, empathize by all means, but don't literally feel the touch experience because you're not being touched or poked. So there's a partial silencing or vetoing of the output of the mirror neuron system so that it allows you to preserve the empathy for the other person, saying feel empathy for the pain that he's going through, but don't literally experience the pain. You don't withdraw your hand when he withdraws his hand. Okay? So clearly something along those lines must be going on. This explains, how do you test this? Well, the prediction is if you amputate somebody's arm, there's a phantom arm, and then you look at somebody else being poked, you should feel that poke in his missing phantom arm. And believe it or not, phantoms have been known for over 150 years. To my knowledge, nobody tried this. So we said, we brought our first patient in, Derek, and we said, okay, we meaning Dave Bryan, Paul McGeough, and I. We looked at this patient with the arm removed, had a phantom arm, simply stood in front of him, and I said, Derek, tell me if you feel anything when I touch my hand. And he laughed and he said, why do you say that? I said, let's try it anyway. So I started touching and stroking and poking my own hand. When I did this, he chuckled and he said he could feel the precise location of touch and poke, and literally feel it and not merely empathize, because he couldn't feel it in his other normal hand, right? And we subsequently did experiments where we poked the other person with a needle, obtained a GSR on this patient. I think we did that, I'm not 100% sure. I think one of our students did that, and showed this person gets a GSR when he merely watches another person being poked, but not if he doesn't. Now, the curious thing about this, which is very intriguing, is one of these patients went home and called me all excited over the phone. And he said, Dr. Ramachandran, you're not going to believe this. I said, what? You know, I have all these pains in my phantom fingers and my phantom arm and my phantom hand. Well, all I do is I go home and I have my wife massage her own hand and palm as I watch. And this phantom massage, which I feel in my phantom hand, relieves the phantom pain. And he was quite certain that was going on. Of course, pain is notoriously susceptible to placebo, so we need to do this in controlled studies. But here is yet another avenue, another approach for relief from phantom pain. Merely watch somebody else being massaged. Okay, now so far I've been telling you about how much time do we have? Okay, we've got 20 minutes. So far I've been telling you about the role of nurture, how malleable functions, the structure and functions of the brain are. Now I'm going to switch to talking about nurture, the role of genes, just to show I don't take sides. I'm going to tell you a little bit about this phenomenon called synesthesia, which was discovered by Francis Galton in the 19th century. He was a first cousin of Charles Darwin. He noticed that certain people in the general population were otherwise quite normal. He thought it was quite rare, but now we know it's quite common. But certain people in the general population had the following quirk. That is, anytime they hear a tone they experience a particular color. So F sharp might be blue, C sharp might be green. But anytime they see a number printed on the page, they see it tinged with a particular color. 5 is red, 6 is blue, 7 is chartreuse, 8 is indigo, 9 is purple, so on and so forth. That every number has a particular color, which is vividly evoked in their mind when they see the number printed on the page. Now, this phenomenon has been known since the time of Galton, as I said, for over 150 years, but has been largely brushed aside as a curiosity or anomaly. And what do you make of somebody who says 5 is red and 6 is blue? Now, so there are several theories floating around to explain synesthesia. Since Galton's time, there have been hundreds of cases reported, but as I said, mainly they've been regarded as anomalies or curiosities. And there have been many theories floating around. Galton also noticed that it runs in families and suggested there might be a genetic basis. We know now that it's true. It does have a genetic basis. And there are several theories proposed to explain it. One theory is, do we have the list of theories? No. One theory is that these people are just crazy. What do you mean 5 is red, 6 is blue? They're just nuts. But one of the things we learn in this business is when somebody is saying something that sounds, that you think is crazy, just think deeper, because sometimes they're telling you something very important. You're not smart enough to figure it out. Okay? But sometimes they are crazy, but it's a different matter. And often it's the case that you're not sharp enough to figure it out. So if you do the right experiments, you can figure out what's going on in your brain. Maybe they'll tell you something about how your brain and my brain also works. Okay. So one theory is that they're just crazy. Well, let's throw that away. Another theory is that they are high on acid or pot, right? And this may be true because it's much more common in California than Minneapolis, and also more common in San Diego State University than here. Having said that, it is true that in certain intoxicated states, certain drugs like LSD can promote synesthesia, but that to me makes the problem more interesting rather than less interesting. Why would synesthesia be more common in drug use? But it's fair to say that many people who never used a drug also have synesthesia, and it does run in families, so that can't be the explanation. The third explanation is that they're remembering childhood memories, that they're playing with refrigerator magnets, and 5 is red, and 6 is blue, and 7 is green. For some reason, they were stuck with these memories, unlike all of us. This never made any sense to me because we all play with magnets on refrigerators which are colored, but not all of us have synesthesia. Secondly, I remember I said synesthesia runs in families. You'd have to postulate that the same magnets were passed down from generation to generation, which again doesn't make any sense. So, but something to bear in mind that of course memory might play a role. The third explanation is more ingenious, and that is they're being metaphorical. As when you say, you all know about intersensitive metaphors when you say cheese is sharp, cheddar cheese is sharp. Well, cheese is not sharp. You can take a piece of cheese, rub it on your skin, it's soft. Why do you say sharp? Well, you say, well, it's just a metaphor, but that's circular. Why would you use a tactile metaphor, sharp, to describe a gustatory sensation? Right? So this raises questions about the origins of metaphors. But saying synesthesia is just a metaphor doesn't explain anything, because what is a metaphor? In other words, in science, you can't explain one mystery with another mystery. Saying synesthesia is just a metaphor doesn't explain a damn thing, because we don't know what a metaphor is. We can define a metaphor, but we don't know what the brain mechanisms are. So as we go along, I'm going to actually turn it upside down and argue that that synesthesia has a concrete physiological basis, which you can pin down in the brain. That in turn can give you an experimental lever for understanding more complex, elusive aspects of the mind, such as what is a metaphor, what is creativity, and so on and so forth. So I'm going to take you on this journey in the rest of the talk, starting from genes, why synesthesia runs in families, what parts of the brain are involved, doing perceptual psychophysics, all the way to metaphor in Shakespeare, all starting with this quirky phenomenon. How do you do that? Well, first we needed to show that synesthesia is a real phenomenon. They're not making it up, that it's a genuine sensory phenomenon. They're actually experiencing these colors. First of all, we found that the phenomenon is much more common than people realized. Ed Hubbard and I did some surveys a while ago, and we found that in fact, it's as common as 1 in 30. 1 in 30 people here have synesthesia. So in this audience, there must be hundreds of you who have synesthesia and it's not come out. So it's very, very common. The most common type being the number color synesthesia. Second most common being tone color or phoneme color synesthesia. So the first question is it real? So we went and polled some students in the audience in our classroom and found 2 students who had experienced number color synesthesia. So we had them come to the lab, and we tested them. We created this very simple display, similar to an Ishihara color plate, same principle, of a number of 5 scattered on the screen. Among them, there are a number of 2s which are difficult to discern. There's a 2 there, a 2 here, should be another 2 there. The 2s form a shape, hidden shape, like a triangle or a square or a circle. The subject's task is to press a button to measure the reaction time, is to find that hidden shape. Now the 5 is red in these guys and 2 is green, or 2 is red and 5 is green. And so the question is, how long does it take to find the 2s hidden among the 5s, the hidden shape? Normal people take 10 seconds, 20 seconds, 30 seconds to discover the shape. If you show it to a synesthete, he says, oh, virtually immediately, he says, oh, I see a red upside down triangle. So if he's crazy, how come he's better at it than us? He does it much faster, at about 1/3 the speed. So this shows we're dealing A, with a real phenomenon, he's not making it up. B, judging from their description, they're saying, I see a red triangle pop out, segregate from a green background. This suggests strongly that it's a perceptual or sensory phenomenon, and sensory processes that are involved in the brain, not high-level cognitive memory associations. Memory associations don't pop out. And Treisman and Beck and others have established this criterion of segregation and pop-out as a diagnostic test for something that's evoked early in visual processing. So having shown that it's a real phenomenon, the next question is, what's happening in the brain? Ed Hubbard and I were looking at brain atlases over 10 years ago, and we were struck by the fact that the brain areas for color and number were coincidentally right next to each other in the fusiform gyrus. That's the dotted green area shows the fusiform gyrus in the temporal. Missing the cursor again. There it is. Fusiform gyrus is there. The green patch represents the color area of the human brain, where colors are represented. Semir-Zeki's V4. The red outline represents the number area. So every time the guy sees a number, a normal guy sees a number region alone gets activated, but a normal guy sees a color, the color area alone gets activated. When a synesthete, these experiments are done using MEG, magnetoencephalography, and initially doing FMR by Ed Hubbard. What happens is, if you show a grapheme, instead of just the grapheme area lighting up, number area lighting up, you get the color area also lighting up in these people with synesthesia. I hope that's clear. So this shows directly, using brain imaging, that there's cross-activation of color by number activation. So what I'm arguing is synesthesia is caused by, just as we had cross-activation between the hand and the face, phantom limb, here you get cross-activation between number and color in the fusiform gyrus producing synesthesia. In other words, we were saying the most common type of synesthesia is number-color synesthesia, grapheme color synesthesia. What's the likelihood they're right next to each other in the fusiform gyrus? Maybe there's some accidental cross-wiring, and we showed that was true by using brain imaging. So every time the guy sees a number, not only is a number neuron activated, but a color neuron is activated, so he sees a corresponding color. Now that was true so we continue to do some psychophysics on these patients, in addition to brain imaging. In some synesthetes, what we have found is even individual numbers have different colors, which is quite mysterious. It again suggests it's not the high-level concept of the number that's evoking the color, but the visual appearance. In this case, even segments of a grapheme can evoke colors in some instances. We also encountered, believe it or not, a colorblind synesthete. Sounds like an oxymoron. They're not monochromats, but had a very restricted range of colors they could see in the real world. But they claimed when they saw numbers and letters, this is not a good diagram, but when they saw numbers like 8 or 9, or 3 or 4, they would see what they charmingly described as Martian colors. That they would see colors in the numbers which they could never see in the real world, physical world. And they couldn't explain this. They said, it's very odd because when I see the numbers, I see these colors, but never in the real world. We can explain it in terms of brain anatomy. The argument is that These people, the retinal receptors are deficient in pigments, so they can't see the full range of colors in the real world. But the numbers allow you to bypass the retina and activate the number region of the brain, and they have synesthesia, the cross-wiring between number and color area, so that evokes a corresponding color, activation of color neurons in the brain. So they see colors, color phosphenes, colors, which they can never see in the real world, so to speak. Therefore, they refer to them as Martian colors or alien colors. This, by the way, strikingly vindicates our argument. Synesthesia cannot be associated with learning from early childhood because you can't learn colors you've never seen. Now, this is true of the first group of synesthetes we met. Everything I've said so far. But then here's another question you can ask. We know that synesthetes see numbers as colored. So 1 is red, 5 is green, 3 is blue, and 7 is yellow, and so on and so forth. What if you show them Roman numbers like V instead of 5? Would they see colors? The answer is very straightforward. In the first 2 synesthetes we tested, they said, no, I know it's a 5, but it's not colored. I know it's 5, it's a Roman 5, but I don't see it colored. Now this is very interesting because what it's telling you is there is not the numerical concept of ordinality or cardinality or numerosity is causing the color, but the visual appearance of the grapheme, which fits the idea that it's visual appearance, because visual appearance is what's processed in fusiform, not the high-level concept of number, which probably happens somewhere in the vicinity of the angular gyrus, somewhere higher up in the brain. Now, I told you that you get this cross-activation in fusiform gyrus in these synesthetes. It turns out this is not true of all synesthetes. There's another group of synesthetes whom we call higher synesthetes or associative synesthetes, who constitute a majority actually. In them, it seems to me that the concept of the number also evokes the color, not just the visual appearance. For example, they'll say even things like days of the week evoke colors. Monday is red, Tuesday is green, Friday is chartreuse, Saturday is indigo, so on and so forth. And months of the year, Wednesday is green, Thursday is yellow. So no wonder people thought they were crazy. Why would days of the week be colored? Why would Sunday be blue? Well, we think we can explain that. What do days of the week, months of the year, and numbers have in common? All of them have in common the idea of numerosity, or sequence, or ordinality. And that's not in the fusiform gyrus. It's probably higher up in the brain, maybe in the vicinity of the angular gyrus. And maybe in these people, in these higher synesthetes, that's cross-activating some other color region, not in the fusiform, some other color region. And therefore, it's a high-level concept of number that's activating the color rather than the visual appearance. Now, so far so good. Now, we haven't tested that directly, but I should mention Ramke in our lab, in our center, visiting us from Amsterdam, has shown there's an actual increase in white matter in the fusiform gyrus of synesthetes. So we've now started, and I remember I said it's familial, so now the next question is why does the phenomenon occur? Why is there cross-activation in these people? Why are there excess connections, if that's what's going on? Well, the answer comes from the fact that it runs in families. So my answer is that in the fetus, in an early infancy, before there's complete differentiation of brain modules, everything's connected to everything. I mean, this is obviously a gross oversimplification, but there's a tremendous redundancy of connections between different brain regions. And as the fetus develops, these excess connections are pruned away to create the characteristic modular organizations of the adult brain, such as V4 for color and the adjacent region for number and so on and so forth, for graphemes and so on and so forth. Now, if there's something goes wrong with the pruning genes, there's a mutation in the pruning gene or genes, this pruning fails to occur, modularity doesn't develop, therefore the adjacent brain regions, which are ordinarily segregated, get linked up together. And you get cross-activation of number to color. So every time he sees a number, he sees a color. So we tested that using brain imaging, as I just said. Now that explains forms of synesthesia and why it runs in families, because the mutated gene is passed down from generation to generation. Now comes the big puzzle, which people have noticed for a long time have not explained. Synesthesia is 7 or 8 times more common among artists, novelists, poets, and other creative people, rather than in engineers and I'm sorry, I shouldn't have said that, but it's much more common among artists, poets, and novelists. Now, why would that be? What do artists, poets, and novelists have in common? What they all have in common is, of course, creativity, but at least some of them, but also the ability to engage in metaphorical thinking or analogy or putting things together which are seemingly unrelated, such as when Shakespeare says, it is the yeast and Juliet is the sun. Well, when you say Juliet is the sun, you don't say she's a glowing ball of fire. You say she is warm like the sun, she's radiant like the sun, she's nurturing like the sun. She's the center of my universe, like the sun is the center of the solar system. You can go on and like that. Shakespeare, of course, is a master at evoking these metaphorical nuances in single phrases. So Juliet is the sun, is pregnant with all of these metaphors. It evokes all these metaphors in your brain. So what I'm arguing is, if metaphors depend on cross-activation depend on linking seemingly unrelated ideas, and if ideas are represented in different brain regions, even far-flung brain regions, then excess connections, if the excess connections, this hyperconnectivity gene is expressed selectively in the fusiform gyrus because of transcription factors, you get this quirky form of cross-activation producing number color synesthesia. But if the same gene is expressed throughout the brain, you're going to get a propensity to link seemingly unrelated brain regions, creating a propensity towards metaphorical thinking. Hence the higher incidence, 7 or 8 times higher incidence of synesthesia among poets, artists, and novelists. Well, people often ask me, if this is true, how come we don't all have synesthesia, if it's so good for you? Well, the simple answer is, synesthesia takes time to evolve. Anything in evolution takes time. So give it another couple of 100,000 years, maybe we will all be synesthetic and creative. But another reason might be that you don't want everybody to be creative or metaphorical. If you're having a neurosurgeon doing surgery on your brain, you don't want him getting metaphorical on you. So you need this heterogeneity in the gene pool. This max of group selection, that's not what I mean. I think you understand what I'm talking about. So to allow creativity to occur, it's very similar to sickle cell anemia gene, where there's a hidden agenda. So what I'm saying is the reason this gene has survived in human evolution, why 1 in 50 people will have this quirk, is because it allows some outliers in the population to be creative and metaphorical. So what have we done here? We've started with I wonder if I can tell them about number lines. I do want to tell you about one further phenomenon. Galton also noticed that, I think I'll skip that and let's go to discussion. We can take this up during discussion. Galton also pointed out that some people had what's called a number line. It's a different form of synesthesia. That is, every time they visualized a particular number, when you and I visualize numbers, you see them approximately arranged left to right. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, going left to right. But it's a very vague propensity. There's no actual, you don't actually see the numbers there. And there's no compulsion to see 1 here and 10 here. But in these people, they have a number line that's vastly more convoluted. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Sometimes even doubling back on itself and going here. It's been a long mystery. It's been confirmed many times since Galton. It's quite common. 1 in 50 people has this. Why does it occur? First of all, is it real? Secondly, why do number lines occur? Why would people see numbers in this quirky manner? I think I can partially answer that if somebody raises this during questions and answers. So let me conclude by saying this is what we do in our lab, in our center. What we do is we take phenomena like synesthesia or phantom limb or apotemnophilia, which has been known for a long time, or Galton's number line for that matter, known for a long time, but regarded mainly as curiosities or anomalies because they didn't fit the big picture of science, brushed under the carpet. We've made it a career out of rescuing these obscure phenomena from oblivion, bringing the patient from the clinic to the laboratory, doing some simple experiments on them, and then find out what's gone wrong in his brain, explain to the patient what's gone wrong, sometimes even develop therapies. Most important of all, what do these findings, what implications do they have for all of us here, not just for the patient? Thank you very much.
Thank you, Professor Ramachandran, for that excellent lecture. At this time, we'll reassemble our panel up front of our other speakers, and we'll start collecting questions from the audience and over the internet. And we'll reconvene very shortly to start.
I heard Martha earlier.
We're going to get started now.
Larger than life behind us.
Good afternoon, Dr. Ramachandran, and thank you. So what we
Good
plan
afternoon,
to
thank
do...
you. This is an unusual experience for me. I've never done this before.
Me either. So what we plan to do is invite the other guest speakers to ask you some questions or contribute some comments, and then we'll take some questions from the audience. So who'd like to go first? John.
So I think I have just a question that gets back to this idea of synesthetes, and that is we know that the... Oh, sorry. We know that they... we know... I was looking at Dr. Ramachandran. Sorry. So we know that you suggest that there's a cross connection that can lead to synesthesia. We know that the face and place areas in the fusiform gyrus, those are areas of the brain that are involved in recognizing faces and recognizing places are right next to each other. Are there syndromes in which people conjure up images of faces when they see buildings or vice versa?
Oh, no, that's a very good question, and it's related to the question people often ask me about why. May you see this? No, you can't see this. It's related to the question of people asking me why we always see number color and very rarely see color number synesthesia. And we don't know how these parameters like numbers and colors are represented in neural networks. It may be the way they're organized. It's one direction is more easy to arrange or more permissible than others. You can have free-floating numbers, but you can't have, you can have free-floating colors. If you have a number, the color labels the number. If you look at a color, where would you see the number? There's something about the way these are represented in the brain that allows only one direction to occur. But that doesn't answer your question. Your question pertains to faces. The answer is yes, it does happen, but it's rare. In fact, the last slide in my show, which I didn't show you, was in fact me having a red halo around my head. And that's because we saw a subject recently, we've now seen 2 or 3 who do see colors around faces. And you can measure this by, you see a red, so this may be the basis of people claiming auras and things like that. So the subject sees a red halo around my face, you put a red spot next to my face. Much more difficult to detect the red spot than detect a green spot. So you can show that it's real, and you can produce interference effects, masking effects, showing these red halos. So the answer to your question, short answer, is you do see face color synesthesia. You don't see face number synesthesia. As I said, it has to do with only elementary sensations can participate in synesthesia. When you start getting into faces and personalities and things like that, it's hard to know what to call it. But one of the things these people say who see face color synesthesia, it's not just faces, sometimes the personality matters too. And this was very puzzling at first, until I realized that the number of personalities is not infinite either. It's a finite set from which you pick. And it's not inconceivable that the fusiform gyrus, in conjunction with other brain structures, also represents aspects of personality, like good guy, bad guy, arrogant, humble, so on and so forth. And this could, in fact, evoke colors in specific brain regions. It gets a little bit speculative.
Hello.
Very fascinating talk. I wanted to expand on this idea of synesthesia and the number of domains it could be in, and whether or not you thought or had seen patients that had some sort of emotional synesthesia. Because I think your comments about metaphors in the domain of depression, which I'll talk about next, is the idea people discuss emotional pain, heaviness. They're actually all states of the cortex, but in fact, you feel like you can't move, but you certainly are paralyzed. And so I think your comments about metaphors might be very relevant to non-sensory systems, and I was wondering what your thoughts were on that.
That's a very good question regarding emotions in synesthesia, because you get casual comments from synesthetes saying that if you put the wrong color with the wrong number, it produces a visceral aversiveness. It looks ugly. And they'll say, Dr. Ramachandran, you look hideous today. You're wearing a green shirt, but your face is red, or some such thing. These kinds of comments are what led people to discard synesthesia as a bogus phenomenon. But they're very valid and interesting observations by these subjects. One specific example is the color emotion, right? It's David Bryan, who's here in our phantom audience here, has studied. Was that color emotion? Right. But every emotion evokes a specific color. You can even produce blends. You blend 2 colors, you get a blending of 2 corresponding emotions. But this is quite rare. You don't see it very often. the links between two elementary sensations is much more common. Why that should be so and why things like face and color and emotion color is much rarer, I don't know, but it does happen.
I'm just going to extend the question then. I mean, all the synesthetic examples you have are areas in very close proximity anatomically. And the question is whether or not maybe are there synesthesias that actually represent very kind of disparate areas of the brain. And given that you have connections, can you have this kind of co-wiring and synesthetic properties in areas of the brain that aren't immediately adjacent?
Well, it's a good question, but it turns out that it's a statistical thing. The kinds of synesthesias you see that are most common, the ones representing adjacent brain regions. But sometimes these synesthesia, but there's no obvious anatomical adjacency involved. So you'd have to argue there are other brain regions where they're adjacent, or that brain regions don't necessarily have to be adjacent to be cross-connected to begin with. There are examples like that. I mean, can you think of an example?
Swimming stuff? Even tone color.
Well, tone color is fine. But, well, we've seen, for example, the man who tasted shapes, you could say the insula involved in taste is right next to S2, which is involved in textures, tactile textures. So there could be tasting of shapes going on there. But I can't think of it offhand. There are many examples where there are distant brain regions where you get synesthesia as well, but much less common.
I have a question. I find it really fascinating that that there is a part in the cortex that's involved in processing numbers. And if you think about evolutionarily, using numbers is relatively recent. And so I wonder what is special about that part of the brain such that now in more recent modern times we use that to represent the image of a number. And you said, Roman numerals don't do it. So how do you think that evolved?
That's a fascinating question. I don't think it's a fusiform, by the way, but I think that's just the grapheme. But I think the idea of sequence may be quite evolutionarily ancient. The idea of sequence, even crows can do it. Sequence and ordinality and quantity, these need to be combined in some ways to create a more sophisticated representation of numerosity that we all humans engage in. Now, where does that happen is a good question. Now, I think the answer comes from, well, first of all, based on clinical neurology and dyscalculias and all of that, I think it's happening roughly in the region of the left angular gyrus. And there's some studies by Dehane and others pinpointing regions of the brain that are involved in representing mathematical concepts and numbers. So the idea of sequence and ordinality and all of that may be represented roughly in the left inferior parietal lobule. But then the question is, are we born with these things or not? I think that we don't have a lookup table for numbers in the brain, obviously, but they're represented, I think, spatially, and whether it's days of the week or months of the year or numbers represented spatially, somewhere in that region. And what I think is phylogenetically, our ancient, we don't have a pre-existing algorithm for doing numbers, which is the point you're making. It's phylogenetically a recent invention, numbers. So how is it done? I think what the brain does is to map, spatial maps on the other hand, are very common in the brain. They go back to the Devonian period, even fish have them. So the brain says the easiest way to represent numbers is not by some abstract algorithm, but actually mapping it onto space, pre-existing space, as on a graph paper. So there's a one-to-one mapping of numerosity or ordinality or cardinality and quantity on a graph paper in the brain, to put it crudely, on the spatial maps. So there's spatial maps of number. And if those goes awry, if something goes awry there, so that this mapping gets disturbed, then you get these convoluted number lines. Otherwise, you can make no sense of why some people would see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. The line even sometimes doubling back on itself. Why would that happen? Only way you can explain it is to assume that the abstract idea of numerosity is mapped onto a spatial map, and something goes wrong with that mapping process, genetically, creating these convoluted maps. And that, in turn, by the way, runs in families, too. It's one of the things we're studying. And many mathematicians often say they have vivid spatial images of numbers represented in space. And they can see hidden relationships between numbers, which most of us normal people can't do because of this disposition in space. Another thing we're studying.
So I want to follow up on this. Oh, I guess Rama, the reason I keep turning around as you're projected behind us. Because I'm very high in oxytocin, I keep wanting to look at the face. But anyway, synesthesia runs in families, and so do some of these other, issues with sort of spatially mapped representations in cortex. Does the familial pattern, is it specific to a pairing of, say, number and color or taste and shape or something? In other words, would a number color synesthete mother have a taste shape synesthete child, or does it breed true?
Well, good question. Well, first of all, statistically, there's a tendency for certain shapes to link to certain colors. So A is more often red than it's blue, for example. First of all, within any subject, it's stable over time. Secondly, across subjects, it can be very different. A can be blue in one, A can be red in another. But statistically, it's more often red than any other color. This may have something to do with the disposition of spatial maps of color and numerosity in the brain, making statistically more likely that you link certain shapes to certain colors. Thirdly, Dave Brang and Ed Hubbard have shown that things with curved shapes tend to be certain end of the spectrum, and things with straight angular shapes tend to be certain other end of the spectrum. Again, showing it's not completely arbitrary, random mapping of colors and shape. There is a propensity in certain directions. That partly answers your question, but was there another question embedded in there which I missed?
Well, I'm just wondering if, these sort of long-range connections between, say, shape representations and taste, or color and number, whether the propensity to make that association, let's see, how can I phrase this? Whether the thing that's inherited is a tendency to make these long-range associations in general, be they between color, number, or face color, or shape taste, or are they specific to a given pair of domains, like color, number? So could, if I'm a color number synesthete, because I assume a given person's synesthesia is restricted to a certain pair of domains, like color, number. So you wouldn't, if you're a color number synesthete, you wouldn't automatically have taste, shape, for example. So, but I'm wondering if whatever genetic predisposition there is to synesthesia is a general predisposition to synesthesia, or is specific to, pairing colors and numbers, for example.
That's a good question. Well, the short answer is there is a general predisposition. Sorry, both statements are true. There is a general predisposition. If you have one type of synesthesia, you're more likely to have a completely unrelated type of synesthesia compared to the general population. That's true. Secondly, if you have one type of synesthesia, your child is more likely to have the same such type of synesthesia than a completely unrelated type. So both things are going on. Hopefully. And also, if you've got tons of connections, you can make you more creative and artistic. That's true too.
Hi, I wanted to ask about tone color synesthesia. So these are auditory cortex and the visual color areas are pretty distant. So it's not working. Is it working now? Yes? Okay. Hello. I wanted to ask about tone color or color tone synesthesia, since the auditory regions that receive frequency information and process tone are pretty distant from the color areas. What do you think is going on there? Why would those areas get connected up? Because this has been reported, the composer Olivier Messiaen, for example, was a tone color synesthetic, and you hear this again and again in the music world. So what do you think is going on?
Well, Ani, you and I did a very extended experiment for about 10 minutes in my lab about 2 years ago to explore these phenomena. We should do that further, given that you are a stone's throw away from our lab. Short answer is not much is known. We've got a new grad student in Miran who's working with us in our lab. And all three of us should get together and solve this problem. But short answer is I don't know.
Well, I'm just wondering if those regions somehow are close together early in the fetal development, and then they get connected up, and that connection persists as those areas kind of drift apart developmentally. well, sounds like something we should work on.
The question is the more central projections of V4, the color area, the higher areas of color, how close are they to primary auditory cortex? And is it conceivable they could be connected, or is there something else going on? They could also be back and forth interactions between different brain areas. I'm giving you a very oversimplified picture of synesthesia. For example, you can show that if you have the letter A, and A is seen as, you know the illusion where you put THE, and the middle letter can be seen as A or H depending on what the flanking letters are. So the color switches depending on what the context is. So it can't just be bottom-up driven, grapheme color evoked in fusiform. Top-down influences also play a role. It's conceivable there's a lot of interaction between auditory and color, which are not just simple cross-activation. But that's a gold mine. We should study it. Mirren is actually sitting here. I don't know if you guys can see her.
Okay. Someone took the bait, Dr. Ramachandran, and asked about number lines. But before we get to that, the most popular question that's come across on the internet, Liz from St. Paul, for example, asked, do phantom limb pain syndromes ever develop on people who were born without limbs?
Now, that's a straightforward question. On rare occasions, I've seen in patients with phantom limbs, first of all, patients born without limbs from birth, most of them don't have phantoms. Maybe 1 out of 10 has a phantom. That's number one. Number two, of those, I would say about 1 out of 10, 1 out of 5 have phantom pain. It's very rare. So I'm saying that 1 out of 100 people with amputation with congenitally missing limbs, they're very hard to find in the first place. When you do find them, they rarely have phantom pain.
So how about number lines?
Sorry, what was that question?
Someone wanted you to please explain more on number lines.
Well, this is something which intrigued me about 12, 13 years ago when I started studying synesthesia. And I was reading papers by Einstein, not that I understood any of it, but papers on Einstein, rather, talking about how he said that he didn't do things in an algorithmic manner in his brain, but would visualize in some sort of space, numerical space. He had a numerical landscape and he would roam this terrain to discover hidden relationships. Many great mathematicians say this. And then I was struck by the similarity between this remark and remark made by people with number lines, Galton's number lines, that they would see numbers occupying regions in space, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, often doubling back on itself. The first thing we wanted to show was, as in our experiments with pop out of color, that this is a real phenomenon. It's not fake. Maybe they're just imagining it. Why somebody would imagine something like that, but leaving that aside, you have to prove that they're not making it up. So Shai Azulai and Ed Hubbard and I did some experiments where we had a subject with a number line where 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and let's say number 93 came back, doubled back on itself and came to number 7 and then branched off again. So in the numerical space, 92 is actually closer to 7 than 8 is to 7 or 9 is to 7. Is that clear? So normally you have a sequence, all of us have a vague propensity to see numbers left to right. So 1, 2, 3, 4, 5, 6, 7, 8. So the numerical distance is not very different from the conceptual, the physical distance of the numbers, not physical, but the phenomenological distance in space between the numbers is not that different from the conceptual distance in numerical space, right? They're congruent. In these people, the spatial disposition of numbers is extraordinarily elaborate and convoluted, but conceptually, you'll find that 5 is closer to 17 than 5 is closer to 2. So what we did was some reaction time experiments. If you ask a normal person, which two numbers are closer, 5 and 2 or 19 and 2? The answer is immediately people will say 19 and 2. You actually do it much quicker, the numbers are further apart. It's called the number distance effect. And there are various versions of this effect. This shows there's an actual analog representation in your brain of numbers, sequence of numbers. Right? So this is an experiment done by cognitive psychologists over a decade ago, 2 decades ago. So in this case, in your number line, convoluted, you can ask, does the reaction time depend on the spatial distance, the geometric distance between the numbers, or the conceptual distance? What determines the reaction time? What we found was it was some kind of hybrid between the two, suggesting that in fact number lines are real in your brain. They are real representations to the extent of affecting your ability to do numerical computations. Now, we haven't nailed this down yet completely, and there's some controversy in the field after we started it, but it's fair assumption that they really have physical instantiation in the brain, and they're real phenomena, just like other types of synesthesia. Question remains, why do you do this? Why do you have a convoluted line? That I already explained in response to a previous question in terms of evolution of brain areas and spatial maps, the requirement of mapping numerical sequence onto pre-existing spatial maps. There's a glitch in this, hardware glitch in this remapping, causing these convoluted number lines to appear. Here again is an extraordinary phenomenon. And why would people see convoluted lines? You can begin to come up with experiments to test this, to show that it's real, and then come up with a reasonable explanation of what might be going on in the brain. Same thing we did for Apotemnophilia or with phantom limb or with grapheme color synesthesia. But here we're not quite there yet. But we've got a student, Nick Root, who's studying it.
We'll conclude with this question. Do any people associate colors with fractions or complex numbers? I think that comes from our math department somewhere. Where are you, Tom?
It's a very good question. That's the great thing about synesthesia. You've got all these obvious questions waiting to be answered. But this is a quick and easy question, quick and easy answer. When they see fractions, they just see each number with its particular color. When I've showed them infinity, they don't see any color. I was hoping they would see square root of minus two as some sort of non-existent color, but they didn't. Irrational numbers, they see non-existent Martian colors or irrational numbers. Short answer is they don't. So yeah, the short answer is you don't see anything really spooky or interesting.
Well, I want to thank you again, Dr. Ramachandran, for your talk and for answering the questions. Thank you.